Thank you to the organisers of these webinar series for giving me the opportunity to talk about some of the really exciting science that we've been doing at Samsara Eco. Each year, we produce 400 million tonnes of new plastic. The overwhelming majority of products that we manufacture from this plastic are by and large disposable. They serve their purpose and they are discarded, usually to accumulate in our environment as pollution with the other 10 billion tonnes of plastic waste already in circulation. We are reminded of our addiction to disposable plastic every day, as waste in our oceans and backyards, as microplastics in our foods and in our bodies. We are reminded that what we are currently facing is a global plastic crisis. This crisis that we find ourselves in is driven by the linear life cycle of the current plastic economy. The life cycle of plastic begins with petrochemical refinement and ultimately ends with deposition in the biosphere as waste or landfill. To confront this, we need a solution to the plastic economy that is circular. We need a solution to plastic management and production that is infinite. This is our vision at Samsara Eco, a future where virgin plastics never have to be made from petrochemicals again. So the management of plastic waste is only one factor that motivates our vision for a circular plastic economy. An often overlooked aspect of the current plastic crisis is the heavy carbon toll that virgin plastic production and current end-of-life cycle management solutions exact on the Earth. Each year, plastic production accounts for approximately 1.5 billion tonnes of carbon dioxide released into the atmosphere. And that amounts to around 3.5% of humanity's total carbon emissions, with some estimates placing this even higher. This has profound implications for our climate, and its consequences only grow with our increasing reliance on disposable plastics as part of a linear plastic economy. The recycling technology that we are developing at Samsara Eco to circularize the plastic economy, in addition to cleaning up our planet, has the potential to save billions of tonnes of carbon, which would otherwise end up in our atmosphere. This is because the current solutions and infrastructures to confront the plastic waste and production crisis are ineffective. Both plastic incineration and pyrolysis, which is the process of thermally degrading plastic at extremely high temperature to liquid oils, are energy intensive and offer only solutions to the physical waste management aspect of the plastic crisis. In either case, the ultimate fate of the plastic is conversion to carbon dioxide, which is liberated into the atmosphere. And neither of these solutions offer any practical approach to extract the chemical monomers for remanufacture of new plastic goods without additional petrochemical refinement. And they therefore continue to only enable a linear and unsustainable plastic life cycle. Bioplastics, which are plastics that are derived from renewable feedstocks such as sugarcane or maize, or plastics that can be degraded by common environmental microbes, also do not offer a circular plastic economy. As with incineration and pyrolysis, the ultimate fate of bioplastics is the conversion, albeit metabolically, into carbon dioxide, without offering any tangible solution to re-extract the chemical monomers needed for the remanufacturing of new plastics. Bioplastics are also hampered by land and water use inefficiencies that are inapplicable to petroleum-based plastics, while offering only marginal improvements in the carbon cost of their production. Like many of the other solutions to the plastic crisis, mechanical recycling alone is insufficient. Mechanical recycling is the primary mechanism of plastic recycling around the world. At the end of its life, only a small percentage of post-consumer plastic waste makes it to a mechanical recycling facility, where if the waste stream is pure enough, it is heated until molten and then reformed into recycled plastic pellets that are used to remanufacture new goods. Because mechanical recycling is a macro process of heating and cooling, with each round of recycling, the quality of the recycled material is compromised a little bit more so that the grade of the recyclate will never match that of the original plastic. With each round of mechanical recycling, the molecular integrity of the polymers of plastic is reduced a little bit more. The chain is shortened. This means that after a maximum of 5 cycles of mechanical recycling, the recyclate will ultimately be fated to landfill as waste, making mechanical recycling much more a mechanism to prolong the lifetime of plastic in circulation before becoming waste, rather than a true mechanism of recycling waste into new material. To minimize the loss of plastic quality during mechanical recycling, recycled material is also spiked with virgin plastic that can only be produced through petrochemical refinement, making the entire mechanical recycling process contingent on the petrochemical industry. The process of mechanical recycling itself is also extremely sensitive to contaminants, making colored, dyed, and mixed plastics fundamentally incompatible with the process. So, if plastic poses such a systemic threat to the stability of Earth's ecosystem and environments, as I've just talked about, why should we continue to invest in plastic-focused technologies instead of plastic alternatives? Like single-use plastics, glass, paper, and aluminium as plastic replacements are at best as detrimental to the environment in terms of total carbon dioxide emissions and energy use. For example, the carbon cost of a disposable aluminium can as a vessel for soft drink is close to 3-fold higher than a plastic alternative per unit of volume. In fact, plastic is seldom the most environmentally detrimental material of glass, paper and cardboard, and aluminium for the overwhelming majority of consumer use cases in terms of total life cycle greenhouse gas emissions. The problem is not plastic, but instead our use of plastic as a disposable entity within a linear life cycle without any efficient or sufficient method to recycle post-consumer plastic into new material. At Samsara, we're using enzymes to circularize the plastic economy. Enzyme-catalyzed plastic degradation offers significant environmental and economical improvements over current end-of-life cycle plastic waste management. For example, some key highlights from an independent study that was published recently found that, and I'm quoting here, enzymatic PET, or polyethylene terephthalate plastic recycling can be cost competitive with virgin PET manufacturing costs. And that the circularity of the PET economy offers sustainability and socio-economic benefits. And this really sort of distills our vision at Samsara Eco, that enzymes can offer a tangible and real solution to the plastic recycling and production crisis that we find ourselves in today. So this gives us a unique technology platform to address the climate crisis while also tackling the plastic crisis. Our life cycle analyses, as well as independent analyses, such as the analysis I just presented in the previous slide, indicate that enzymatic recycling of plastic at scale can displace significant global greenhouse gas emissions while circularizing the plastic economy and allowing for the remanufacture of virgin quality plastics infinitely. Here's how it works. Our process is tolerant of contaminants such as dyes, allowing us to take feedstock of any quality, even that which is rejected by mechanical recycling facilities. Our platform enzyme technology breaks the plastic down to its fundamental chemical monomers. Unlike mechanical recycling, which is this macro process of heating and cooling, Samsara recycling is molecular. Our enzymes break the chemical bonds that make the plastic polymer up and liberate the chemical monomers that make up the plastic. This is, for example, terephthalic acid and ethylene glycol in the case of polyethylene terephthalate or PET plastic. Our process is efficient, sustainable, and economical. Because plastic is remanufactured by the extension from the chemical monomers produced by our platform enzyme technology, the recycled plastic material is virgin quality and there are no limits on the number of times a given piece of plastic can be recycled by Samsara without compromising the recycler. The process is truly infinite. We can even take the chemical monomers produced by our enzymatic recycling process and upcycle them into value-added chemicals, giving access to larger and more diverse markets. Altogether, our platform technology means that we can accept the cheapest, the most accessible plastic feedstocks, those which are rejected by the mechanical recyclers or have hit their end of life in terms of mechanical recycling. We can break this down into chemical monomers in an efficient process with a reduced carbon footprint and produce the highest quality products, all while being completely disconnected from petrochemical refinement. This all, of course, hinges on having enzymes that are able to break down plastic. While the number of extant enzymes capable of breaking down plastic discovered in nature is constantly growing, they are often restricted by the same biophysical limitations. A common and consistent theme among PETases, these enzymes that catalyze the hydrolysis of PET plastic, is consistently low activity. The catalytic turnover of these natural proteins and the resultant poor mass transfer when they're applied to industrial processes is often too low to be economical at industrial scale. They are also plagued by low thermostabilities. The enzymes unfold and lose catalytic competency at temperatures around or below those that industrial plastic recycling processes need to be run at, imposing a higher catalytic loading on the process, greater catalyst consumption by the process, and frequent catalyst replenishment and production. This poor thermostability also manifests as poor recombinant protein yields, resulting in higher energetic and material costs to produce industrial volumes of recombinant enzymes, and further decreases the efficiency and viability of processes that are centered on and built around natural plastic degrading enzymes. The solution to these challenges is to engineer better, more efficient, and industrially useful PET or PET degrading enzymes. While this is very easy for me to sit here and say, enzyme engineering can be incredibly challenging. And today I'm going to be talking about some of the ways that we have been using enzyme engineering, specifically evolution-guided enzyme engineering and rational protein design, to design thermostable, catalytically competent, new-to-nature PETases with industrial usability for the infinite recycling of PET plastic. So I want to begin by talking a bit about evolution-guided enzyme engineering. As the name indicates, evolution-guided enzyme engineering leverages evolutionary information to produce novel, highly functional enzymes. This, of course, must begin by understanding the processes of evolution. We can describe evolution as descent with modification along a phylogenetic topology or tree. In this example that I have on screen now, which is the phylogenetic tree of animals or metazoans, we can see shared ancestry as descent from common ancestral nodes or bifurcations at the interior of the tree. These bifurcations represent extinct ancestral species. For example, the last common ancestor of vertebrates, which existed several hundred million years ago and diverged into extant jawless fish and jawed vertebrates, is represented by the internal node that separates these taxa out. Likewise, the first member of the animal kingdom to emerge, which existed on the order of 1 billion years ago, is represented by the last common ancestor shared by Cnidaria, which are jellyfish, and the vertebrate group, which is right here at the root of this phylogenetic tree. We can study and understand the molecular evolution of enzymes in the same capacity by studying these phylogenetic trees. Instead of the tips of this phylogenetic tree representing different taxonomic groups, they represent different extant enzymes. Likewise, the internal nodes of the phylogenetic tree that I have on screen as a sort of arbitrary example represent the extinct ancestral enzyme sequences that extant proteins have diverged from. For example, an enzyme that catalyzes some arbitrary chemical reaction A recently shared a common ancestor with a homologous enzyme that catalyzes a distinct chemical reaction B at this node here within the phylogenetic tree. We can directly translate this evolutionary information into sequence and biophysical novelty using ancestral sequence reconstruction, or ASR. If each tip of a phylogenetic tree represents an amino acid sequence, we have a phylogenetic topology that describes how these amino acid sequences have diverged from one another. And we have a statistical model that captures the molecular evolutionary process. We can computationally reconstruct the extinct ancestral sequences that represent the bifurcations in evolutionary history. This process of ancestral sequence reconstruction can be essentially imagined as extrapolating the observed sequences back in time along a phylogenetic structure or topology using a predefined sequence evolution model. Ancestral sequence reconstruction can provide us with insight into the mechanism by which proteins and enzymes evolve. It is tremendously useful in allowing us to understand the biophysical and molecular features that enzymes or proteins have to acquire or lose to adapt their phenotype. But it is also profoundly useful for enzyme engineering. Ancestrally reconstructed proteins often have novel phenotypes that can make them extremely useful in biocatalysis. For example, a widened or altered specificity profile that is distinct from those found in extant proteins, or improved thermostabilities. In this somewhat arbitrary example, we might expect that the last common ancestor shared between these groups of enzymes that catalyze reaction A and reaction B may have had an intermediate phenotype that isn't completely reflective of those in group A or group B, or something entirely different. So, in a sense, ancestral sequence reconstruction allows us to transform evolutionary information into sequence space, where we can explore for new-to-nature enzymes that have improved biophysical properties over natural extant enzymes. This diagrammatic figure sort of summarizes the process of ancestral sequence reconstruction. It begins with the generation of a phylogenetic tree or topology, which is a search through tree space using some sort of tree search heuristic. And we ascend this kind of likelihood gradient until we produce a topology that maximizes the likelihood of producing the observed data that we have, our sequence information. Through ancestral sequence reconstruction, we can translate this phylogenetic information into new sequence space that we can explore for functional enzyme sequences. As an extension of ancestral sequence reconstruction, we have developed a pipeline that we term multiplexed ancestral sequence reconstruction. Like conventional ASR, multiplexed ASR transforms evolutionary information into sequence and biochemical novelty. However, we explicitly incorporate evolutionary uncertainty to produce tens of thousands of new-to-nature and functionally enriched sequences that encompass multitudes of concurrent evolutionary hypotheses. The important thing to note about our method is that despite our best estimations and most sophisticated parameter-rich sequence evolution models and phylogenetic tree search heuristics, we cannot ever know the topology of a phylogenetic tree with complete certainty, and that's because it's retrospective. These phylogenetic structures have evolved over billions of years of enzyme evolution, and we cannot really know that with complete certainty. Multiplexed ancestral sequence reconstruction translates that into a full evolutionarily accessible sequence space that we can explore for highly functional and new-to-nature enzymes for biocatalysis. So like the figure that I just sort of talked about that describes ancestral sequence reconstruction as being centered or pried on a single phylogenetic topology, our approach takes multiple incongruent distributions of phylogenetic tree space that are equally likely at representing the data that we have observed, and translates that into a much larger and much more diverse sequence space that we can explore for biochemical novelty for protein engineering. So evolutionarily guided enzyme engineering strategies, to summarize, give us two major advantages in protein design. The enzymes they generate are synthetic. They explore sequence space outside of what nature has provided in extant enzymes and proteins and allow us to identify novel sequence fitness peaks that have been unexplored by evolution. Now, I'm going to talk about this in a little bit more depth shortly, specifically in relation to our novel PET degrading enzymes. But I wanted to highlight the utility of our enzyme engineering design approach in kind of exploring sequence space beyond what nature has given us. Because of systematic biases in ancestral sequence reconstruction algorithms, including those that we use for multiplexed ancestral sequence reconstruction, the enzymes produced by our approach also typically demonstrate thermostabilities that are elevated above what would be expected in natural enzyme counterparts, meaning that industrial processes built around these proteins can be run hotter and for longer with lower catalytic loading and less frequent catalyst replenishment, making them ideal for industrial biocatalysis and already addressing several of the issues that I raised previously in regards to the industrial application of natural petases. So our first mission at Samsara, as I have alluded to before, was to engineer enzymes that can break down PET or polyethylene terephthalate plastic. PET is one of the most commonly used plastics globally. We all know it as the main ingredient in our disposable plastic water bottles and our disposable food containers, but also as polyester in textile material. As I've also kind of alluded to previously, many natural enzymes have evolved the ability to break down PET plastic. These enzymes typically belong to a group or a family of enzymes called cuteinases that are functionally involved in degrading plant cutin, which makes up part of the plant cuticle. And cutin is this hydrophobic ester that is chemically quite similar to PET plastic. So it's with these natural cutinase enzymes that we wanted to begin our PETase engineering campaign. This is what our enzyme design pipeline looked like. We start with an initial computational design step where we use multiplexed ASR to generate libraries of functionally enriched enzymes that we believe are promising PETase candidates. We have the DNA encoding these libraries synthesized and cloned by our friends at Twist into expression vectors before transforming them into lab-strain microbes, expressing and purifying the designed enzymes, and ultimately assaying them for their ability to break down PET plastic. Our PETase assay involves directly taking PET plastic, cutting it up into discs, and incubating it with our enzymes at an appropriate temperature to replicate the expected industrial conditions that the enzyme may be deployed in, before directly measuring the breakdown products of PET plastic, which are monomer equivalents of terephthalic acid, primarily terephthalic acid, but also some species such as M het and B het. And we do this by absorbance. We iterate on this and incorporate the information that we've learned into the next round of evolutionarily guided designs. So we began our experiments looking at the enzymes that have previously been identified as petases, these cuteinases. And as expected, we found several lineages of cuteinases that possess significant petase activity. Specifically, many of the cuteinases belonging to bacteria from the genus Thermobifida, as well as the leaf compost cutinase and its close homologs. So leaf compost cutinase is really kind of this anomalous enzyme that was identified by metagenomics in hot leaf compost heaps, basically, to identify enzymes that might be useful industrially at breaking down hydrophobic polymers. And it kind of belongs on its own evolutionarily away from these other Thermobifida-related cutinases. We can see that, consistent with many other studies that have come before, the leaf compost cutinase-like cutinases are significantly better than petases that belong to the Thermobifida cutinases. So we can see here in our distribution of phylogenetic topologies that we use for multiplexed ASR, that we have these families of cutenases, specifically those belonging to the genus of Thermobifida that all cluster around here, as well as the leaf compost cutenase-like enzymes down here at the bottom. And, we can clearly see that those that are related to the leaf compost cutenase are significantly more competent petases, specifically this enzyme here, which was only somewhat recently characterized as being a very productive, fast, natural petase. And this is particularly true at higher temperature. These petases, the leaf compost cutenase-like petases are significantly better. We have also included a previously published engineered variant of leaf compost cutenase, which at the time was one of the fastest petase enzymes. And we indeed find that it is a significantly faster petase than any of the natural enzymes that we have identified or screened. So as part of our evolutionarily guided enzyme engineering, we used multiplexed ancestral sequence reconstruction to reconstruct a library of ancestral sequences that capture the evolutionary uncertainty that we observe in the cutenase family phylogeny, this distribution of kind of competing yet equally likely phylogenetic topologies or trees that describe how these extant cutenases have diverged from one another. Before I start talking about the activity of these enzymes, I want to point out that they are truly novel sequences. Here I have used a large protein language model, which is a deep learning model trained on the full protein universe of sequences. And these models embed sequences as high-dimensional vectors that capture their evolutionary and biophysical properties. And in this sense, these representations are an empirical representation of sequence space, projected down to 2 dimensions so that we can comprehend them, but they show, informed space of the sequence landscape that we're investigating. So the closer 2 points are in this space, the more evolutionarily and biophysically similar the sequences they represent are. And we can see that from our extant library of cutenases, we can sample regions of sequence space that have been unexplored by nature through multiplexed ancestral sequence reconstruction. These represent complete sequence novelty, accessible only through our evolutionarily guided enzyme design strategy, and really exemplify why we use these types of enzyme engineering approaches to explore beyond the natural diversity of extant enzymes. So in this graph, we can see that the extant cutenases that we have investigated are shown in blue, and the ancestrally reconstructed sequences that we use are shown in salmon. And while there is a lot of cases where there is overlap in the sequence space that these 2 different populations of cutenases sample, there's also a lot of sequence space that is accessible only in the ancestral sequences, where the natural enzymes simply haven't explored that space yet. When we characterize a small library of our ancestrally reconstructed proteins, within the very first round of designs, we found petases that are significantly more active than even the fastest wild-type leaf compost cutenase-like enzyme. So here in blue, we have wild-type extant petases, including 1 or 2 engineered variants for industry. And in salmon are the first round of our ancestrally reconstructed proteins. We can see that, the activities of some of these enzymes match or exceed those of the fastest leaf compost-like cutenase enzymes, and even some of the engineered variants. We can see this as well by looking at the raw data, these absorbance traces, where again, the extant enzyme leaf compost cutenase is shown in blue, and our fastest ancestrally reconstructed variant, ancestor 547, is shown in salmon. In the regions where PET monomer equivalents absorb between 230 to 270 nanometers, there is a significantly higher concentration of PET monomers that are liberated by ancestor 547 than there are by leaf compost cutenase under the exact same experimental setup and circumstances. What's really remarkable, though, is that this design process, thus far at least, was completely blind. And what I mean by that is that there was no manual intervention or rational mutations or selection required. We tested only a really small library of 48 variants, and our design pipeline was able to blindly generate this repertoire of quite distinct enzymes that matched current industry state-of-the-art PETase activity. These enzymes were all generated and produced by evolutionarily guided design pipelines, exploring sequence space outside of the natural diversity of proteins without us having to rationally put any sort of requirements on that. So if we look at where these ancestrally reconstructed enzymes that are competent active PETases emerged in cutinase evolution, we find something really interesting. Our two most active ancestral variants, Ancestor 547 and Ancestor 442, belong to equivalent ancestral nodes in distinct phylogenetic topologies. This node is where this node is the most recent common ancestor of leaf compost cutinase and thermobifida cutinases, which are both groups of extant active PETases. But we can see that the tree topologies that either of these ancestors are sampled from are incongruent. So what I mean by that is that both of these phylogenetic trees sampled the same sequences. We have our thermobifida-like PETases here and our leaf compost cutinase PETases here. But the structure of these two phylogenetic topologies is different. The placement of how they were expected to have evolved and what groups are ancestral to each other is incongruent, even though the most functional ancestors are still sampled from the same ancestral interior node of that phylogeny. What is really interesting about this, though, is that we sampled this equivalent phylogenetic node, this most recent common ancestor of leaf compost cutinase and the thermobifida cutinases, from at least several other phylogenetic topologies, which produced cutinases that were inactive PETases, but still expressed as soluble protein. This tells us that the different phylogenetic contexts that are provided through our multiplexed ancestral sequence reconstruction pipeline make significant contributions to the diversity of phenotypes we produce in our ancestral enzymes. Had we sampled only a single sort of best guess phylogenetic tree without accounting for any opposing phylogenetic hypotheses, such as these two here, it is very likely that we would have completely missed this PETase fitness peak in sequence space entirely. And it really sort of demonstrates the importance in our design strategy and significance that sampling this evolutionary diversity contributes to our overall engineering of PETases. So having generated competent PETase backgrounds through multiplexed ancestral sequence reconstruction, we next turned to a different enzyme engineering approach. Oh, is this going to play? And this is rational protein design. Now, the principle of rational enzyme design is, as the name indicates, to make intentional rational mutations that perturb the enzyme structure to invoke a change in its biophysical or catalytic properties. Unlike design strategies such as evolutionarily guided enzyme design, which are blind as I described before, successful rational enzyme design requires a really intimate understanding of how the enzymes being engineered work, how it interacts with its substrate, and how its structure relates to its function. One of the tools we have to understand this relationship between structure and function is molecular dynamic simulations. And this is a video that I'm playing now of one of our molecular dynamic simulations. And we simulate the motions and dynamics of a biomolecular system to understand the basis for its activities. In this example I have on screen now, there is a model of one of our designed enzymes, which is in blue, and it's interacting with a short chain molecule of PET plastic in black, white, and red. We can see how our enzyme interacts with the PET substrate in the substrate binding groove. And we can make targeted mutations that promote a tighter binding between these two molecular species and therefore lead to better and more productive catalysis. This also highlights one of the advantages of being partnered with the Australian National University, which is that we have access to resources such as the Gaudi supercomputer that allow us to conduct these computational experiments, which are incredibly resource intensive. And that also extends to our multiplexed ancestral sequence reconstruction pipeline, which also requires a lot of computational power and resource to perform efficiently. So an example of a rational design that we found to increase the activity of our petases is this phenylalanine 211 that protrudes from the PET binding cleft and sterically occludes the PET molecule from the site of catalysis. Mutating phenylalanine 211 into a smaller residue significantly improves the activity of these enzymes. Interestingly, this specific mutation was also found to significantly improve the activity in the background of LCC, or leaf compost cutinase. And this was published by other researchers a few years back, and it sort of highlights it as an Achilles heel in PET degradation across different groups of diverse cutinases. It also shows us that evolution is nowhere near perfecting natural petase enzymes. Because plastics have only existed for the last half a century, which is a blip in evolutionary time, the enzymes that break it down do so largely through promiscuity, that is, unproductive side reactions that happen to occur due to chemical similarity between substrates that are not under selective pressure. And this is really sort of an example where a really competent petase is only one mutation away from a cutinase, and that there simply hasn't been strong enough selective pressure or a long enough evolutionary time for these mutations to become fixed. Again, sort of emphasizing why there are lots of problems associated with deploying natural petase enzymes in industrial processes. So after several rounds of rational design, we were able to improve the specific petase activity of our ancestrally reconstructed cutinases by over an order of magnitude, according to our mid-throughput assays. Here we have our final petase designs in red and the extant cutinases, petases, and previously engineered petase variants in blue for context. And it's clear that there is a significant improvement in the throughput of PET degradation over the wild type and extant enzymes. There's not really any sort of comparison here. It's clear that our ancestrally reconstructed and then further rationally designed enzymes are vastly superior petases at breaking down PET plastic. Additionally, because our assays were conducted at high temperature to replicate the industrial conditions that the enzymes would be exposed to at scale, the enzymes we have selected are also thermostable, and they're produced in high recombinant yield in lab strain microbes. And this really sort of addresses almost all of the challenges associated with enzymatic PET depolymerization that I presented and talked about earlier in the presentation, as to why we can't use natural enzymes for PET degradation and why that process is simply inefficient and uneconomical. So we can take these enzymes, purify them to homogeneity, and confirm that outside of our medium throughput assays, they indeed do continue to degrade PET plastic as determined by HPLC instead of absorption. What's more is that using an identical plastic substrate from the same manufacturer, Goodfellow, who manufacture these rolls of amorphous PET plastic, the rate of degradation by our engineered enzyme significantly exceeds those of recently reported engineered petases. We can also show that our enzymes are only marginally slower at degrading post-consumer thermoformed PET plastics, such as this biscuit tray, than they are at degrading amorphous manufactured PET plastic, which again shows that these enzymes are useful industrially. They are able to take post-consumer waste and efficiently break them down into the monomer equivalents of PET. The most remarkable thing about these enzymes, though, is that they are completely unique. They share only distant homology with other engineered or characterized petases. In fact, the closest characterized homologue to any of our repertoire of petases is leaf compost cutinase with only around 65% sequence identity, which is well and truly outside of the immediate neighborhood of homology. Ancestor 547 shares at the absolute most around 75% sequence identity to any other protein sequence in any database, characterized or otherwise. And we can see this when we embed all of the cutinase sequences that we've investigated and characterized into one of these large protein language models that transform amino acid sequences into high-dimensional vectors. The enzymes which are most active, Ancestor 547 and its designed descendants, belong to completely new to nature fitness peaks that are distinct from the petases that have emerged in nature. So we can see here in this plot where I've colored the sequences embedded in this model by their activity, that the petases we've produced that are the most competent, the fastest, the best at degrading plastic, are also completely new to nature. They don't really resemble any other extant protein, and they don't group together with them in these high-dimensional spaces. This highlights the power of our technology platform to rapidly produce truly new to nature novel enzymes with catalytic properties that are ideal for the industrial degradation of post-consumer plastics. And we're expanding this platform. We're expanding it to confront the plastic crisis in all of its forms by developing enzymes to degrade and break down and recycle all post-consumer plastic wastes. And this will accelerate us towards our 2030 ambition of recycling 1.5 million tons of plastic waste per annum at Samsara. So this project represents a tremendous amount of work from the whole Samsara team. This is a picture of us all together recently at the Australian National University, but I think since then our team has grown significantly. But the science that I've talked about today was done all in partnership with the Australian National University and represents a lot of hard work and dedication from Colin Jackson, Vanessa Vongsuthi, Rosie Georgelin, Jennifer Ton, and Brendan Lee. Thank you for all of your attention. Again, I'm really excited to have been here to talk about this research to you all, and I would be happy to try and answer any questions I may be able to. Thanks.